Health is often reduced to symptoms and treatments, but much of it is shaped by unseen processes within the body. In this conversation, we explore how balance, disruption, and everyday choices shape long term health and well being. Joining us for this conversation today is Dr. Vikrant Bhor.
So before we begin with the interview, would you please introduce yourself in a few words?
So, hello everyone. I am Vikrant Bhor. I am scientist self and head Department of Molecular Immunology and Microbiology at the ICMR National Institute for Research in Reproductive and Child Health. I'm also an associate professor at the Faculty of Medical Research at Academy of Scientific and Innovative Research. And I just like to put in a disclaimer that all the views expressed herein are my personal and do not reflect on any thoughts or ideas of my organization.
So then we'll move on to the questions.
Sure.
Our first question for you is that you have had a fascinating career path from your early work in the Indian Institute of Science on Metabolic Pathways to your long tenure at ICMR. So, in this way, how has your understanding of the microbial world shifted as you moved from looking at a single bacteria to the complex living ecosystems of the human microbiome?
So, I mean, I think that question kind of encompasses my entire journey, like where it began and where I am right now. So, I actually did my bachelor's in microbiology. So when we began studying microbes initially, as it happens generally at the undergraduate level, there was a lot of emphasis on single organism and association of organisms with disease. So medical microbiology was my favorite subject, and I would really look forward to every day learn and explore new facets of pathogens. We were also exposed to the beneficial organisms, but the view that we have currently that both have to work in sync was not really existing in those days. So I mostly viewed uh, you know bacteria and mostly as those ones which are really causing a lot of disease and harm. And there was a lot of emphasis on how do we study, explore these, and see whether we can come up with any management or treatment strategies against these. So this is how the view existed when we started out, and it persisted during my post-graduation right up to the time I took up my postdoctoral research. So that was the opportunity I got to really do an in-depth investigation of a particular pathogen, that was Mycobacterium tuberculosis. So there again, the entire view and the overarching aim was to explore the pathogen. A particular, we were working on the biotin biosynthesis pathway. So explore the metabolic pathways to find out whether these can serve as drug or vaccine candidates. And that's what we did. But gradually, as I moved over in my career, in the year 2012, when I was faculty at NIRRCH, I happened to take a coursework module. And in that coursework module, while teaching or rather preparing for the lectures, I came across the fascinating concept of microbiome. So that was the time that the human microbiome project was, at least the first initial findings were released. And I found it extremely fascinating. The fact that you no longer talk about microbes as single entities, but as a community, as a network, both the ones who have characteristics which are considered beneficial and the ones who are not so beneficial, all working or forming a network and governing all our physiological functions. So that idea, that concept came very late. And then onwards, it has been really, really fascinating. So once that idea really caught my fancy, I think I shifted all my attention, and the focus of my work became the microbiome, and it continues to remain so.
That's really nice, sir. Now, moving on to our second question. During your time at Piramal, you worked on anti-diabetic and anti-obesity agents. How did that experience in the private sector drug discovery shape your current approach to developing probiotic therapies for public health challenges like the neonatal cholestasis?
Yeah. So there again, I think it was you know, just my good fortune. I've been working on projects which were drug discovery related. Throughout my postdoctoral research at IISc, and then the second stint at Curie Institute in Paris. But there is a difference in the academic setup and the one you have in the industry. So this is not to say that it's not professional enough, but in the industry, while I was at Piramal, I experienced what it takes to put a molecule, uh, you know, to develop a therapy, not just identifying a chemical, but or characterizing it, but what is the process that goes before and after, right? So it's not just the work that one does in the laboratory, but the whole thought, the idea, why should one choose a target? So doing a SWOT analysis and then deciding whether this target is good enough to take or work on, and then uh, finally developing assays to finding molecules, to finding inhibitors, etc., and then doing the animal testing, etc. So it's a whole process. So I went through the whole process, and then though the molecule that I was working on did not really move in that direction, there were other examples which I observed very, very closely. So that really, made me, made my ideas more fixed. And then right now, but when I entered NIRRCH, when I came to NIRCH, we were working mostly on basic research projects. So until recently, all our findings were more of exploring the pathogenesis and mechanisms. But of late, we have started, formulating projects which are more translational in nature, and all my experiences, that I had undergone in Piramal and other places are really helping me, make, you know, formulate such projects. So let's hope. I mean, the hope is that eventually we'll get some, come close to making a product.
So that was wonderful to hear. Our next question is based on your experience in applied and translational microbiology. What are the major regulatory challenges in translating microbiome-based research into effective and safe therapies or dermatological therapies, to be specific?
That's a really pertinent question. That is where actually, more focus or attention is required. So I'll just quote, when we are right now, we are thinking of developing microbiome-based therapy, and there are some already there, but the regulatory environment is not really, you know, very streamlined right now. So the path is not very clear, especially in our country, because other countries, I mean, FDA is approved two therapies for um, some ailments which are, so this is for fecal microbiota transplantation. So fecal microbiota transplantation has been carried out um, in several places, and there are a lot of instances. There have been trials, etc., which have been done, but it has not come. It did not come directly as a approved therapy. A modified version of it in the form of live biotherapeutics was approved by FDA, and we have two such FDA approved therapies right now for ulcerative colitis and Crohn's disease. But the path that has been taken abroad, I would say it's the same has not really happened right now. There are several factors, I would say. I mean, for example, quoting the case of FMT, if one has to do fecal microbiota transplantation is nothing but the transfer of fecal matter from a healthy donor to a diseased individual, just as you would do blood transfusion or blood or any transplantation of any organ. So that actually is something that is done when most conventional therapies or approaches fail, right? So, but to do this, one really requires a good infrastructure. So right now it is being done on a one-off case, an experimental basis, or where everything fails. So it is not really come as a therapy in India, and our regulatory guidance is, I mean, there's no regulatory path as such. In fact, there are articles which have been written about it. So we are facing whether it is FMT or any other microbiome transplantation that we are currently thinking, it is really, really lacking. So, and then how do we go about? Because without that, researchers like me would be stuck, and all those people, aspiring researchers like you or your colleagues would also be stuck. So the way forward, I think, is somebody has to. We are taking initiatives, at least creating an environment, holding discussions with experts, and taking their view how to go forward. Because it's clear that such therapies are really required, and they are happening elsewhere. They should also happen here in our country. But then right now it seems to be a slow process. But then we are taking some steps, and hopefully in the near future we may have some such therapies, or at least some clarity about how to go about.
Yeah. So moving on to our next question. How feasible is it to integrate microbiome-based interventions into the mainstream healthcare, and what obstacles currently limit their large-scale adoption?
So, I mean, this is in continuation with what I already said. So right now, microbiome is kind of a buzzword, right? Everyone knows or has heard about the microbiome, and it seems to be like the magic bullet. If everything you can just pin it down to microbiome, and microbiome is involved. So I would say yes, microbiome is a big component. Uh, but sometimes I feel we also tend to exaggerate the claims. Nevertheless, it is a very, very important thing, and it serves a dual purpose. Like, it can also be of predictive value, whereby it can help in predicting the state of health of an individual. Like, just as you have a blood count and blood parameters, the microbiome also the microbiome composition, especially the gut microbiome composition, can indicate the person's health. Though it cannot be viewed in isolation, you have to really correlate with all the other physiological parameters. So, what has been observed is there are a lot of startups which are focusing on that, whereby they're doing diet-based intervention. So you know, uh, I mean, they've come up with tests whereby they do the microbiome composition, and based on the microbiome composition, they are advising some sort of dietary interventions. So this is all common sense. Like, I mean, if we make changes in our diet, even without knowing anything about the microbiome, if you have a fiber-rich diet or a more diverse diet, it is going to. So, but this knowledge is expanding, and so this is one aspect. Okay, the diagnostic aspect, or where it helps us in making changes. Similarly, microbiome in severe conditions or even chronic diseases, if one knows the microbiome of an individual at the beginning of a therapy, and later on, if you find out at the end of the therapy, probably there can be correlation to find out whether the microbiome composition can predict how soon an individual, or how well an individual will respond to treatment. So these are the kind of applications that are, I'm just elaborating on the applications, and then, coming to the hurdles. So that is one aspect. The second is where you actively take microbiome, you modify or modulate the microbiome. One is where you are, I just said, where you are looking at the microbiome composition and then trying to derive meaning out of it. And the second one is where you know that the microbiome is not proper, or there is dysbiosis, the composition is haywire. Now you want to modulate it so as to achieve a better state of health. Sorry. So when it comes to the predictive or diagnostic aspect, I don't think there are much challenges, though there are no approved tests so far. It is all happening in a very informal manner. Uh, so then there is a lot of knowledge, but scientifically documented, evidence in the form of, say, trials, clinical trials is missing. There are hardly any trials, I mean, very few trials, if at all, which are being conducted. So therefore, we are not really having a very proper scientific evidence, which is really required to translate any new knowledge, any new therapy into practice. And like I mentioned, there are regulatory gaps right now. So all these are, no, I would say bottlenecks, but they are there for any new therapy. It's not really, you know, and the other aspect is it is very generalized. I mean, it can be applicable to everything. Like, for example, if you talk about cancer therapy, or I mean, or monoclonal therapy, etc., you know that what is the ambit of that. But microbiome is such a general topic, and the fact that microbiome is applicable in almost every sphere of life, be it the plant kingdom or the animal kingdom. So therefore, it makes these things complex. But I feel the regulatory environment has to improve, and that would be better if more and more people are doing scientifically generating scientific evidence in the form of the highest evidence, which is trial. So I think slowly we will move towards it, but right now it's not as per our requirement.
Now, going deeper into your area of research, how central is the gut microbiome to overall well-being beyond digestion, especially in terms of immunity, energy levels, and inflammation and other areas we don't take into account?
Yeah, that's, I mean, I would say it is as central as, I mean, I wouldn't call it the like a brain, but it is no less than the brain. And of course, all of us are aware of the gut-brain axis. So the gut is a very, very critical. I mean, going by the fact that it has the longest surface, it just does not serve the digestive, the function. There are a lot more things it does. So the gut or the tissue underlying the gut also harbors the largest reservoir of immune cells, right? So when you know that there are so many immune cells in the underlying tissue, and you also have so many microbes in close proximity, the connection becomes obvious. So the microbes, one of the biggest applications or their influence is on our immunity. So I would call it a very, very primary or a dominant form of influence, because apart from digestion, so if the microbes exist, they have, first of all, a lot of diversity in their genetic makeup. So therefore, they have a huge repertoire of enzymes which can practically do anything, right? So right from digestion to clearing of xenobiotics to, so they have a huge potential. So the fact that they are existing, they can contribute to all these things. But the metabolites or the products which are generated as a result of these activities are the ones which are really, really important. So the metabolites which are produced, to quote or name a few, like short chain acids, indole, etc. These are short chain fatty acids, etc. These are very, very integral. So they, in fact, interact with our immune cells on the surface of the immune cells and trigger signaling pathways which are tied in to various functions, changes in immune function. For example, they can trigger an anti-inflammatory response through increased generation of regulatory cells. Some of them may also lead to increased inflammatory pathways like in cytokine production. So by that way, we are influencing immunity. Now, immunity and digestion are in two aspects. As I mentioned, it also, these metabolites don't just stay localized, within the gut and of course, they interact with the underlying immune tissue, but there is also through the vagus nerve, they connect with our nervous system. And then there is a gut-brain axis. In fact, right now we are, we know that there is a gut-liver axis, gut-lung axis, and so many axes are being discovered. So what are these axes? So it is nothing but the gut metabolites, the metabolites produced by the gut bacteria, which enter into the circulation and find their way into these organs. And by entering these organs, they are able to interact with the cells in those particular organs, the tissues that make up those organs, and the cells, and then they influence their signaling and overall function. So they have really, you know, actually can go anywhere. And I mean, this is not just the internal surfaces, the skin, for example. I mean, all of us, next to the gut is the skin microbiome. The skin microbiome is, of course, very important. So, I mean, simple things like if you keep washing your hands too often, or you keep your dry, it also causes problems. So, I mean, there is a certain microbiome composition which is necessary for functioning of the that particular organ or system, and that is required. If you wipe it away, be it through practices like frequent washing or ingesting antibiotics or anything which is not favorable, it is bound to cause a problem. So in this manner, I mean, it is really, and I mean, there is also something I guess probably we can come up talk about it later. But right now, I would just like to say that these are, in a nutshell, the processes that the microbiome influences.
So, like you mentioned about the treatment options and the applications of the microbiome, continuing on the same line, the treatment strategies like probiotics and say microbiome-based therapies, when they develop, how do you decide whether we're restoring the natural balance or changing how the body normally functions? Like adapting to these therapies, are we adapting to the therapies or are we changing the entire balance of the body?
Uh, so if especially when we talk about microbiome, so there is this concept that every individual, or rather in a complex or living system such as the human body, there are niches of microbiome, right? Every particular organ system, or rather the surfaces, have their own microbiome composition. So over time, right from childhood to the, as we advance, there is a certain community which has developed, and the interaction of the members of the community, the network that they form, is necessary and essential for the proper functioning of that system. It is not, this is not to undermine the role of the cells which are there, but then their functions are augmented, regulated through these microbial metabolites. So when these are present in different I'm sorry, I lost track of the question. I was what was your question?
So when we talk about therapies, like treatments like probiotics and microbiome based
uh,
therapies, how do we decide
yeah.
whether we are restoring the natural?
Correct. Correct. So therefore, like I was getting there. So when you have these networks already established, so so these are mostly resilient. Generally small changes, there are a lot of changes. Like say, if you go from one region to another, or if you eat something, you are ingesting new microbes, or people from as they travel, or whenever there's any change, there is going to be a small change, or perturbation in the composition, right? But the overall microbial community is resilient, doesn't disappear or change drastically. So there is something called functional redundancy. So even if there are minor changes at the species level, if there's a change, there are other microorganisms which can step in and probably complement or take over the function of the lost microbes. So there is this kind of a redundancy and resilience. But when there are large changes. For example, when I say when we take antibiotics, now antibiotics are, unfortunately, antibiotics many of them are broad spectrum, so they are not really directed only against the pathogen. They would have a wide action on all the microorganisms which are present over there, and then this leads to wiping out certain microorganisms, right? And those could be the beneficial ones. So this leads to a state of dysbiosis. That means the balance is disturbed. Right? So most of the times, these perturbations, there are numerous perturbations, drastic change in diet, change in sleep pattern, change in location, any kind of stress that you, or a sudden change and a continued and prolonged kind of a change is going to disturb this balance. So therefore, most of the times, the right way would be to restore the balance, right? But right now, what do we have in the form of therapy? We have largely monotherapy or therapy in the form of a couple of microorganisms which are put together and they are administered. So, what would happen? The danger of this is that when you take these, I mean, most of the time they just overwhelm and uh, push out the pathogen, and then the microbiota of that particular location recolonizes. So this should happen gradually. But many a times the fear is that when they stay there and they overgrow, they may change the composition altogether. And these could be long-lasting changes. So therefore, the best way would be to use therapies which are maybe a mix of pre and probiotics. These are called symbiotics. So when you give prebiotics, they create a favorable environment for uh, the microbiota to establish themselves. Without that, if even if you give a lot of them, they not probably may not be there or colonize. So right now, the therapies that we have, there's a lot of variability in their efficacy, and that is because not all of them are able to adhere and colonize the surfaces that where they are supposed to be. So therefore, the efficacy of all these treatments is not uniform, right? So we need to really come or go towards treatments, therapies which are guided by the ability of the microbes to colonize a particular niche. So these kind of, so that I think if we work in that direction, probably we may be able to restore in a proper manner rather than just give some arbitrary kind of treatment, which will be like just uh, a sudden kind of a response which may not have sustained efforts, sustained results.
So now, based on your work, moving on to the next question, if you could change one thing about how women's health is approached clinically through microbiome research, what should it or would it be?
So this is the heart of our work right now. We are focusing on, so just as there is fecal microbiota transplantation, there is also therapy or a proposed therapy called vaginal microbiome transplantation. So ideally, where there are conditions where the microbiome is really uh, completely deranged, and then there are these recurring conditions, including recurring infections, etc., where despite all conventional treatments, one does not really achieve the kind of results, and it also fuels the constant administration of antibiotics and other available therapy constantly fuels antimicrobial resistance. So there's a acute need for coming up with such you know, revolutionary or what I would say game changer kind of applications like the vaginal microbiome transplantation. Having said that, it is not very easy. I mean, if you think about it, it is taking the vaginal secretions from a healthy donor and then uh, giving it to individual who has a very completely deranged microbiome. So now this, where would you find this kind of a deranged microbiome? It could be antibiotic resistant infections, recurrent infections of the reproductive tract. Uh, there may be also some individuals with infertility. Now, they are not really responding to treatment. And if you go and really check, dysbiotic microbiota may also be associated with such a condition. And there could be other more severe conditions like cancer, cervical cancer, vaginal cancer, and so on and so forth. So there's a range of conditions where there's a need to really come up with adjunct therapies. So not to say that chemotherapy or any other form of therapy is not there right now. There are no drugs for these infections and cancer, etc. But uh, what is really required is to really uh, push the therapies so that if you can adjunct or supplement these therapies with something, maybe we can achieve a lower dose of the chemotherapy or the antibiotic, and then it will lead to less resistance. So this is where I think we want to work. So vaginal microbiome transplantation is one such area that we really want to work on, and we are making efforts towards that. Of course, it may not always be direct transplantation of the vaginal secretions, but it could be in some other form of any other product, etc. So this is what we are. So given an opportunity, I think this is uh, the kind of contribution that microbiome science can make towards improving women's health.
I think that's really interesting. So, moving on to our last question of this round. What question about microbes do you think today's science is still not brave enough to ask?
I mean, there are so many things we know and they are very obvious, but we fail to really. Uh, I mean, it's like, I mean, knowing everything but not really acting on it. So it's common sense. Like, I mean, and I feel what microbiome science has just told us what we already know, right? But what we fail to do, or I mean, I'll give you a few examples. I mean, why talk about science? It's life and life practices also. The fact that when you are undergoing any treatment or therapy, you should possibly have a probiotic included along with it. So this kind of a practice is not there. So despite extensive knowledge and documentation of these obvious facts, we are not really following these practices. So uh, I would say scientifically there are many challenges. It's not as easy as we make it out to be. But uh, where we also lack is the fact that we know about it, but we are not able to really put it into practice. So a lot of advocacy is required. Uh, uh, and of course, I would say that this is again not a magic bullet. You can't just do change the microbiome and or rather have some therapy and forego any other kind of therapy. So that is there. In terms of scientific question, I think we are exploring everything. I mean, I think there's hardly any area using microbes as decoys or using them to for targeted cancer therapy, etc., or even very chronic. So we are doing everything. We, as in science has reached a point where there's a lot which is being done. So scientifically, I think we are uh, challenging so many norms and uh, old ideas, and we're moving forward. Like it was kind of unheard, like a therapy like FMT. If you tell somebody that you have cancer, and then probably you can take a fecal transplant and hope to get better. This was like kind of very alien. Or rather, a weird kind of thing. Can it really be? An individual who is already immune compromised, and you're trying to give more microbes. So this was, the kind of speculation, rather ridicule that those people who started such therapies would have undergone. But science has changed a lot, and there is a lot of evidence which is being generated where you can really modulate a microbiome in a very drastic manner to achieve better therapeutic efficacy in many disease conditions. So I would say we have been asking the right questions, but implementing what we know is not really happening very fast.